Hello, we've made it to week 4 where we're going to be talking about cells and the cell structures. We're going to start by doing a quick overview of prokaryotic cell structures. So in week 1, we were really introduced to this idea of the hierarchy of life and putting all of life into these different categories, where we started with all of life, thinking about every living organism on the planet that ever existed now or in the past. And of course, there was lots of different ways to classify and to characterize, particularly at even these highest levels throughout history, where, of course, originally things were separated into plants and animals, basically. Um, then we found out, no, there's some more things that we need to get into there. Now we're working in the domain structure where we've got the 3 domains we talked about: bacteria, archaea, and eukarya. So the relationship between these eukaryotes and prokaryotes is that prokaryotes are the bacteria and the archaea. The eukaryote are the separate, that we'll have a different video to go over the cell structures because that's a lot more. So this is one way of looking at the evolutionary path of all of life and how things are related, where we can see that archaea are more related to eukaryotes than they are to bacteria. The exact same graph, slightly different graphic, you see the exact same thing. We have these prokaryotes that we're putting into one category, that's the rest of this PowerPoint, and then we have those eukaryotes, and we all come from the same ancestor. This graphic, I absolutely love this figure. It shows the timeline of life as a road trip. So instead of having time as time, it's a distance, where you have the origin of Earth up here 1.6 billion years ago, and then you have it going all the way to Boston, where humans finally are. And the reason why I'm putting this slide here, this is really a huge part of Bio 182, is just to realize. So it took a little bit of time for Earth to cool down, to have some solidifying rocks and such. But so that took a little while, but really life began quite early on, within a first like billion years or so. And then for a significant amount of time for life on Earth, it was only the prokaryotes. So if you're driving all the way to Boston, prokaryotes from San Francisco, all the way up to Oklahoma. What is the point of me saying this? Prokaryotes rule this Earth. We might not see them, we might not think about them, but numbers wise, you are even more made up of cells that are prokaryotes. You got 10 times more of their genomes in your body and on your body than you do eukaryotic cells, which actually make up that are holding your DNA. Okay, so we've got the 2 major categories of cells, again, not domains, but categories, where we've got these prokaryotes and the eukaryotes. And what we're going to see is that prokaryotes are more simple. Again, we've learned from week 1, they do not have a nucleus. They do not have little special membrane and bound organelles. You can also see, first picture, prokaryotes are much, much smaller than eukaryotes. But there are some common characteristics of all living cells: a plasma membrane, which is going to, for the prokaryotes, go around, and then there's the fluid inside, and that's the same. We call it the cytosol if it's a prokaryote or a eukaryote. Both types of cells are going to carry their genes or their genetic material in chromosomes, but they're going to have slightly different structures in prokaryotes and eukaryotes. And then they need to take that information in nucleotides and convert it into the stuff that makes up the cell, and that's proteins. And so that's done physically. The location is in the ribosome. So we've got our prokaryotic cells. Again, we're just diving in a little bit more to focus in on some of these structures that the rest of this PowerPoint is going to go over. But these are really what you need to know. So if you already know what all of these structures do, if you can recognize them by a picture and tell me their function and their purpose, you are golden. I will review those now, though. So prokaryotes we can divide into 2 categories. We can divide into the non-photosynthetic. So these are bacteria and archaea that are going to be getting their energy from eating sugars, not by ingesting them, but by taking them from the environment and bringing them into that plasma membrane and using those sugars to get their energy. Whereas there are also photosynthetic bacteria and archaea that are not going to need to get sugar from elsewhere, but can make it themselves. They can make it themselves primarily from using sunlight. And again, we have an entire chapter on that later on in the class on photosynthesis. So what do you need to know for prokaryotes? You need to know prokaryotes do not have a nucleus, but they still have genetic material. They still have DNA, and that's going to be in a location. The location it is called the nucleoid. And you're going to see for eukaryotes, we also have the word nucleus and we have the word nucleolus. And if you're dyslexic like me at all, these words all look almost identical. So you need to be very careful about which one is which. A nucleoid is in a prokaryote. It is not a nucleus. We've got ways for these bacteria to move. There's pili and there's flagellum. We are going to be talking about flagellum again in eukaryotes. So it looks like the same type of structure. Functionally, they have the same purpose, but physically how they're made up, totally different. Prokaryote, totally different structure. But both the pili and the flagellum are used for movement. The pili are like these little tiny hairs that are going to be moving, where the flagellum is like a long whip. Um, Ribosomes, as I already mentioned, ribosomes, learn this now. You're going to need to know it in much more depth on week 6-ish, I think. Ribosomes are the site of protein synthesis. So all cells take that genetic material and they convert it into proteins that do the stuff in the cell, and that is done physically at a ribosome. So those are demarked here and here and all over here. Um, Again, we're going to see eukaryotes also have ribosomes, but again, their structures are a tiny bit different. The size of the proteins that make them up are different. The plasma membrane. The plasma membrane is the most interior of this membrane of all these lines, and it's really important. We're going to have a whole chapter next week, actually, chapter 5. We're going to talk about the plasma membrane and how it can let certain things in and other things out, and doesn't let other things in, and how that all functions. So no, it's really important for deciding who comes in and who comes out. It's like the bouncer at a cellular nightclub, is how I like to say it. Outside of that plasma membrane is like a cell wall. And the cell, well, it's not like a cell wall, it is a cell wall. And what that cell wall is like is if you kind of picture a chain link fence right here, where it's got a lot of holes in it, but it gives it a hard end that you can't keep going past. So it's a hard end where that cell can't expand too much, because it has that hard structure. And this is going to be really important when we're talking about how water is going to be moving into and out of a cell. The plasma membrane cannot burst open in a prokaryote that has a cell wall, because it's going to be that harder structure that's going to give it a backing. If we were in class, I would walk up to the back of a wall and I would show you how I can't keep walking because the wall physically prevents it. On top of that, not all prokaryotes, but some prokaryotic cells have on top of that cell wall, they'll have a capsule, which is often sticky, full of carbohydrates, full of sugars that can be used for attachment. They're also great at protecting the cell, protecting the prokaryotic cell from our host immune responses, particularly cells called phagocytes, which we're going to learn about in the future too, which come up and eat a bacteria cell that's infecting them. But a big giant capsule that's all like sticky goo makes it very difficult. Again, I mentioned this already. The cytosol is a little bit hard sometimes for students who aren't really focusing, because you go, what is this pointing to? It's pointing to nothing. Exactly. The cytosol is the fluid. The cytosol is all the fluid that everything is in. It's going to contain salts and other molecules, but it's just the fluid, so it's the thing pointing to nothing. And then lastly, we have a chromatophore. Chromatophore are parts of the plasma membrane that have been infolded. Now, these are only going to be in photosynthetic bacteria or prokaryotes, and that's because this is where photosynthesis is taking place. So if you don't do photosynthesis, you are not going to have any of these infoldings of the plasma membrane that contain all those proteins. We're going to see in the eukaryotic cells, we're going to refer to these as the thylakoid membranes. And we're going to see how they're related to these prokaryotic cells. So very lastly, you should now have an idea. No, you should know these facts about where DNA is found, how is the cell bounded, what kind of membrane-bound structures exist there, larger, smaller, multicellular, and then some examples. So I'm not going to physically read this all, but other than saying, again, the definition of a eukaryotic cell is that it has this nucleus, it has these membrane bound structures. And that is it.